Hello everyone and a warm welcome also from my side. My name is Schafer, I am team coordinator at Miltenyi Biotec, and today I want to talk to you about ultra-high content imaging and how it helps to identify novel target candidates in the setting of pancreatic adenocarcinoma. When we look on the pancreas on a cellular level, we can see that you can roughly split it in two parts. One is the endocrine system, whose task it is to produce and secrete hormones, most famously known insulin, produced by the beta cells, which stirs our blood sugar levels. The grand majority of the cells, however, is formed up by the exocrine system, here in yellow acinar cells and in blue ductal cells. Acinar cells produce digestive enzymes, which are then transported via the ducts into the digestive system. It comes to no surprise, as the acinar and ductal cells form around 90 to 95% of the total cell mass in the pancreas, that also the majority of pancreatic neoplasias is derived from these kind of cell types. If a neoplasia arises from one of these cell types, we call it a pancreatic ductal adenocarcinoma, or short PDAC. It is a very dreadful disease and has a median 5-year overall survival of only 7%. PDAC develops, as mentioned, from the acinar cells that undergo acinar to ductal metaplasia, or from the ductal cells themselves. In most of the cases, mutation starts with the KRAS mutation that has been shown in murine models to be sufficient to form precursor lesions. However, this mutation alone is not sufficient to cause PDAC. For this, we have the accumulation over several more mutations throughout the years that lead to increasing desmoplasia and aggressiveness of the cancer. The current treatment options split up depending on the stage of the tumor when the patient presents themselves. Around 20% of the patients have a resectable tumor at the stage of diagnosis. In this setting, we can reach a median overall survival between 54 months in the latest clinical trials, while standard of care is more 35 months. Another 20% of the patients have a borderline resectable tumor. In this setting, we have a 20 months overall survival. Unfortunately, these patients can undergo surgery, but from the resectable patients, 90% relapse afterwards again. The grand majority of the patients presents themselves in a metastatic stage. Here the median overall survival is 11 to 6 months old. The main pillar of a treatment of PDAC until today is chemotherapy, and the main chemotherapeutics, 5-fluorouracil and gemcitabine, are in use since decades. There are a few experimental targeted therapies, however they are only eligible for small populations of the patient. A new hope have been CAR T cells. They showed unprecedented efficacies in B-cell malignancies, and the treatment algorithm looks like this: that we extract a blood sample, normally leukapheresis, we isolate T cells, modify them genetically, then we expand them, and in the end reinfuse them into the patient. A CAR stands for a chimeric antigen receptor, and it consists normally of an extracellular antibody binding domain, a spacer region, transmembrane domain, and intracellular we find co- and main stimulatory signaling domains that try to mimic the naturally occurring T-cell receptor signaling. The natural way for a T-cell to recognize a tumor cell would work via the T-cell receptor. The T-cell receptor is able to recognize mutated peptides which are presented via the MHC complex. This is not possible in the first place for several reasons, like the immunosuppressive tumor microenvironment, expression of suppressive molecules of the tumor cells, downregulation of the MHC complexes, self-antigens, and so on and so forth. Thus, the CAR T-cell strategy looks like this: that we took an antibody-based screening in order to identify tumor-specific surface antigens. Once identified, we can take the antibody binding domain, draft it onto our CAR T-cell, and with this we generate a new specificity of our CAR T-cells towards the tumor cells. The aims of the study are split up in two parts. The first part deals with the identification of specific and safe candidates. I think it goes without saying, but nonetheless, if you have a target antigen which is shared between healthy and malignant cells, we will have equal disintegration of the healthy and malignant cells, with a side effect that may be bearable for B-cell malignancies, as B-cells are a dispensable cell type. However, this is not the case for most of the targets which are currently under investigation in the setting of solid tumors. The second part will deal with the generation and evaluation of target-specific CARs. As depicted with this figure, you can see that the CAR consists of several subdomains, and in the meantime there are a myriad of domains available that you can incorporate into your CAR framework. Thus you do not know a priori which one is the most effective one. And this has to be determined experimentally. The first step on our journey to identify specific and safe target candidates was conducted by the screening of 2 patient-derived xenograft models. We dissociated them and screened via flow cytometry around 400 surface markers. We identified 105 target antigens to be expressed on these patient-derived xenografts, and we screened another 15 patient-derived xenografts, all with different mutational backgrounds, these 105 markers, and identified 50 target candidates to be expressed on these. Thus, the screening revealed markers that are specific on patient-derived xenografts. Until now, we only investigated the expression of 50 target candidates and showed that they are specific to patient-derived xenografts. But we have no idea until now how their expression looks like offsite the tumor site. How is the healthy tissue expression of these 50 candidates? In order to find this out, we performed screenings from in silico databases. We used 2 RNA-seq databases, 2 mass spec databases, and 1 antibody-based database. And we ranked these 50 target candidates according to their off-tumor or healthy tissue expression. We ended up with the prioritization of these candidates, meaning that the highest one has the lowest off-tumor expression and vice versa. In order to verify the results that we were able to generate on patient-derived xenograft models, we wanted to verify this now on primary human PDACs. And for this, we took advantage of the newly developed cyclic immunofluorescence platform, now called Maxima, and it works as follows. On the bottom right side, you see an immunofluorescence image with a 3 marker overlay. And this is a pretty standard thing, and a lot of laboratories can do this all over the world. It gets, however, a bit difficult when you want to have another marker stain, say a 4th or a 5th. And if you want to have a look on even more markers, this is even more difficult. Maxima resolves this issue in the following way. We perform a staining on a tissue section. We then have take an image. We erase the signal. We perform a new staining, take an image again, erase the signal, and this cycle continues over and over and over again. To enable you to get a rough idea of the power of this tool, I brought for you this slide on which you can see all the images taken by the run in a 3 marker overlay. We have always in blue DAPI and in red AppCam, and in green is the respective marker indicated in the upper right panel. This Maxima run was consisting altogether of 97 markers, 4 of which were not able to stain nicely. The other 94 you can see here at one glance, and you can see that it's now easily able to multiplex this different marker expression on the very same tissue section. You can do it also by yourself by following this QR code, or just Google Maxima R tool. There you will find the opportunity to click and press the markers on this PDAC tissue and find it out by yourself. We took this to investigate the following. In green you can see cytokeratin, which is in this instance a pan-epithelial marker, and we know we're looking here on tumor cells as we have by microscopy the morphological features that you do not have, for example, in flow cytometry. And we were looking now in red that the marker of interest, like here CD59, is co-expressed with the tumor cells, and this is not the case for this marker. This is the case for CLA, for example, where you have green, the marker of interest is nicely co-aligned with the tumor cells. Also co-expressed, but to a much lower extent, is CD318. As you can see here, the red color is less bright, and please keep this in mind because it will become important on the next slide. After having now verified which markers are expressed on primary human tissues using the Maxima tool, the next step was to add some statistical power by increasing the number of tissues analyzed. And for this we took the 8 highest ranked candidates, which were the most specific as revealed by the Maxima analysis, and we took now again primary human tissues, but now instead of using them for a Maxima analysis, we dissociated them similar to the patient-derived xenograft models and analyzed them also in flow cytometry. The results are depicted here, and you can see that all these markers are pretty specific to EpCAM positive cells. I talk now here in purpose from EpCAM positive cells, as in contrast to a microscopy technique. In dissociated tissues, we cannot be really sure if an EpCAM positive cell is also a tumor cell, as the morphological features of this cell are missing. In addition, it happens that if you get a tissue from a surgeon, the surgeons normally aim at cutting out more of the actual tissue than would be theoretically necessary in order to be really on the safe side, to have taken out all of the tumor cells so that nothing of the tumor stays remnant in the patient. This leads now, of course, to the following situation, that you may have a mixture of tumor and healthy cells. Thus, if you're gating here only on epithelial cells, you cannot be sure to 100% that we are dealing with tumor cells. And for example, you can see that T-span8 and CLA are highly specific towards these epithelial cells, but on the other side, such a target like CD318 shows a rather heterogeneous expression on these cells. For some instances, only 20% of the supposedly tumor cells being EpCAM positive, you find this target expressed. This can be explained by another fact. I've just showed you before in the Maxima that CD318 has a dim expression as compared to other targets, as for example CLA. And this is now also verified here in the flow cytometry analysis. And the analysis and gating strategy of a dissociated tumor tissue is far from being trivial. You have a lot of debris, autofluorescence, and then when it comes in addition to a target candidate or a candidate of interest like CD318, which is dim expressed, it depends on the gating where you set it and the overall looking of the tumor, how many positive or negative cells you get. This is less a problem for other target candidates. For example, here CD66C, T-span or CLA, you see they are very bright on all the epithelial cells, and this is not the case for CD318. In fact, this analysis that I brought here with me is a pretty nice tumor. You have often cases that this is not such a bright staining, and this is why you end up then with negative cells, although all the tumor cells are really specific for this target candidate. It is also not by chance that I show these 4 target candidates here. It's CD318, CD66C, CLA, and T-span8. They are the top 4 target candidates from our screening, as they had the highest bioinformatical safety ranking. They have the highest specificity to the tumor cells, and this is why we took them for the further course of our project. Of course, the in silico methods that we used screening these databases does not really tell you something of the real expression within the tissue, but it's rather a ranking within a certain set of candidates. Also, RNA sequencing do only have a certain predictive value, but not always does RNA-seq confirm results that you would then gain by protein expression analysis. Also, you have the disadvantage that you cannot dissect between different cell lineages within an RNA-seq bulk sequencing or a mass spec bulk analysis. And this is especially with pointing out to a CAR T-cell therapy of utmost importance, as you may get a signal within a certain tissue, but the signal derives from a set of cells that are dispensable, and thus we wanted to perform an experimental antibody-based analysis method on healthy tissues, and the results are shown here. You see here the 3 target candidates, CD66C, T-span8, and CD318, we have 16 healthy tissues, and in the upper left panel is always a pancreatic cancer tissue. And now we looked on the expression of these target candidates in these healthy tissues. You can see for CD66C it seems to have a low expression in the lung. Here you have a green spot in the ovary, but I can tell you that this is a cell empty space. We have here a situation where a mucus is secreted in an empty space, and thus we have antibody binding but not towards cells, so this may most likely be not a big problem. Within the skin, CD66C seems to be expressed in hair follicle cells, which may be a dispensable cell type in immunotherapy. TSPAN8 has a strong expression within the colon. This is worrisome with respect to off-tumor toxicities in a CAR T-cell therapy and should be considered carefully. CD318 has, if at all, a very dim expression in some tissues. As a next step, I want to mention that immunofluorescence analysis is to some extent always dependent on the investigator and actual settings on which you use during your analysis. I can tell you that all these images are taken with the same settings, so you can really compare the intensities between the pictures. Yet you can make a negative signal positive and the other way around by playing around with the parameters, and thus we were aiming for a more objective way of analyzing our samples. For this, we used from the tissue sections that I just showed you and laid a bioinformatical analysis over them. We identified cells and then we analyzed or let the program analyze how strong the expression on the respective cells are for these target candidates. And you can see that the pancreatic cancer tissues, always depicted on the right side, have a much stronger expression again of the target candidates as compared to the healthy tissues. Some findings as before, like that CD66C has low expression in the lung, or that in the colon we find a lot of TSPAN, holds true also in this analysis. Important finding is that the expression within the tumor is always much stronger of the target candidates than in the healthy tissues, and also interestingly, for example, TSPAN is expressed in the colon, but the other target candidates are not, which would again increase the safety when you're aiming for a combinatorial approaches. To sum this first part up, we were able to identify 4 target candidates, namely CLA, CD66C, CD318 and TSPAN8. They have a high tumor specificity and a restricted off-tumor expression with a high and confident bioinformatical safety level. If these target candidates are really safe depends also to some extent on the CAR function, as you can play around with the certain parameters there. You can tune the affinity by changing the SCFV or tune the effect by exchanging other domains, and thus we aimed at evaluating this now empirically. Thus, with these 4 target candidates, the next task was now to perform CAR cloning, and we chose a setup as follows. When we look on the CAR, we have the same framework throughout all CAR constructs with respect to the transmembrane domain, co-stimulatory signaling and main stimulatory domain. This stayed the same. We had difference only in the length of the spacer, and depicted here is the shortest spacer used, that is derived from the IgG4 molecule and contains only the hinge between the CH2 and CH1 domain. We have also a bit longer one derived from CD8 alpha stalk. We have an M-sized spacer, which is derived from the CH3 domain of the IgG4, and a long spacer, which is derived from the CH3, CH2 domain. And as I meant, we have to test this, as we do not know a priori where the binding site of our antigen-specific antibody is, and as we do not know how long the target molecule is, and it has been shown in literature that you can fine-tune your CAR effectivity with the spacer lengths, we wanted to find this out experimentally. Also, a difference can make the orientation of your SCFV, and we also switched this, ending up altogether with 34 CAR constructs that we now tested in vitro assays. We were looking on marker expression, cytokine release, and cytotoxicity. What is an effective CAR? I brought two videos with me today, and I would like to ask you to first have a look on the right hand side where you see the co-culture of tumor cells depicted in green with CAR T cells. You can see that the CAR T cells are able to fast and efficiently kill all the tumor cells, while on the left side the mock control, which is untransduced CAR T cells co-cultured with the tumor cells, are not able to kill all of the tumor cells, and the one that survived the first few hours do survive to the very end of the experiment. This case is pretty clear. This is a nice and effective CAR. However, this is not always the case, as you can see here. In gray you see again the mock control, and in violet you see the very short spacer targeting CD66C, and this seems to be ineffective in this assay. You have a few CARs that show intermediate efficacy, and you have two CARs that show high efficient killing. We also analyzed marker upregulation on the CAR T cells, for this instance PD-1 or interferon gamma release, and what we found normally is that the activation and cytokine release goes well in hand with the killing efficacy of the CAR T cells. Again, it became clear that different spacer configurations, like here the XS and S, do have different efficacies, or here the XS and S, and this is something that needs to be considered and that we found out. You may have recognized that I only showed results for 3 target candidates. This is caused that CLA, the other target candidate, is a self-antigen on CAR T cells. It is proposed to be a glycosylation of the protein backbone CD162, and when you have T cells isolated from peripheral blood, around 20% of them express it. But once activated, the expression increases throughout time until after 7 days of activation, around 80% of the T cells express this. This poses a technical roadblock and needs to have some technical adjustments in order to overcome this problem. For the other 3 target candidates, we were able to identify suitable CAR candidates. For T-SPAN8, we had 2 equally well performing CARs, and for the other 2, 1 each. As a next step, we wanted to test our CAR constructs now in vivo. And here you can see the results with the mice treated with untransduced mock T cells. As you can see, the mock T cells are not able to halt the tumor growth in any regard. On contrary, after 12 days, all the mice have to be sacrificed as the tumor starts to ulcerate. When we look on the CD66C treated group, we see that these CAR T cells are able to hold the tumor growth and control it, but they are not able to reduce the actual tumor burden. This is the case for the long T-SPAN8L spacer, but in a heterogeneous way. We have 2 mice where we see a tumor hold and control, and we have 3 mice where the tumor mass is shrinking. It looks a bit more homogeneous with the T-SPAN8S spacer CAR, where all the mice show tumor eradication, but with a rather heterogeneous response. For some mice it takes a bit longer than for others. And for the CD318 CAR, this looks a bit faster, stronger and more homogeneous, very effective. So we can say that indeed the best performing CARs in vitro are also functional in vivo. Next, we wanted to investigate, again using the Maxima tool, a bit closer what may be a potential reason for the dysfunctionality of the CD66C CAR. For this, we compared a tumor from a mice treated with CD66C CARs as compared to a tumor treated with CD318 CARs. In the first panel, you can see at one glance that at the bottom part you have a lot of cyan and violet cells, meaning you have a high infiltration of CAR T cells. In the upper panel, you see a lot of green cells, which are mock T cells, so untransduced T cells. For whatever reason, CAR T cells did not infiltrate or stay at the tumor site. We find a lot of tumor cells left in the upper tumor, which is logical as the T cells did not engage some cytotoxic activity. And in the bottom panel, there are only but a few remnants left of the tumor. A very interesting finding in my eyes is that the bottom tumor is also flooded with mouse macrophages, which is remarkable in that regard that we use NSG mice, which are immune and not immune competent, and the macrophages in these mice are believed to be dysfunctional. But we see a clear difference here between the tumor treated with a functional CAR, where it is full of mouse macrophages, while in the upper panel, a tumor treated with a dysfunctional CAR, we have only a few macrophages in the closed perimeter of the tumor. We can exclude that target downregulation is a potential mechanism for tumor escape in the upper panel. We see here a lot of CD66C still nicely co-expressed with all the tumor cells, and we see also still some expression of CD318 in this tumor, but as mentioned a few times before, CD318 is dim expressed, so this is not so bright as for CD66C. We could also verify the results for CD66C in flow cytometry. In the bottom you see the unstained control with some background, and here you clearly see that CD66C is still expressed to a high extent on these tumor cells. Thus, the decrease or the decreased function of the CD66C CARs cannot be explained by target downregulation. With this, I'm coming to our final summary. The successful generation of CAR constructs and in vitro testing was performed. We could determine the best in vivo CAR constructs for 3 target candidates. All 3 target candidates, or the CARs, therefore showed cytotoxic efficacy ranging from tumor control to complete tumor eradication. The dysfunctionality of the CD66CS construct remains unclear, and for CLA is a self-antigen, and some further development needs to be undertaken in order to find technological adjustments to enable us to generate successful anti-CLA CAR T cells. If you want to reread what I just showed you or want to know a few things more in detail, you can do so, as these results I just showed to you have been recently published in Nature Communications. Just follow the link as here in the bottom part if you are interested in this topic. With this, I want to come to the conclusion and also the outlook. The discovery and evaluation of 4 novel target candidates was conducted in a field of a high medical need. This is in fact the first study with a holistic and also comparative target analysis in the field of PDAC, and in my eyes it is nice to have an integrative approach between different techniques like RNA-seq, mass spec, overlapping here with antibody-based flow cytometry and cyclic immunofluorescence staining. In fact, enabling us to have a broad idea about the expression of certain markers in certain parts of the human body. This study as such provides a novel comprehensive target discovery algorithm, and I have the hope that this might be interesting and also followed and of course also improved by others, because here I have the hope that this may us to enable better and more targeted therapies and thus help patients in the future. Next steps would be to evaluate combinatorial safety measures for the novel targets. As I mentioned before, maybe one could make use of so-called AND CAR strategies or not CAR strategies, or adapter CAR system. It would also be interesting to benchmark or combine the novel targets with already known targets like mesothelin, which is currently the target with the highest interest in the field. And last but not least, the development of a technical solution to overcome a CLA CAR T cell problem, for example a CLA knockout, would be feasible to generate CARs for this target. Here are my references, and this is the disclaimer. Finally, I have a lot of people to thank. In fact, I'm pretty proud that I am a representative of the work that was conducted, obviously not just by me, but by a lot of excellent friends and colleagues. And for this, I want to thank our collaboration partners from the University Medical Center and MPI in Göttingen, Frauke Alves, Philipp Ströbel, Janine Misbach-Güntner, Diana Pinkert-Leitch and Sabine Wolfgram. I want also to thank our colleagues and collaboration partners from Charles River, Julia Schüler, Melina Lamorte and Dorothee Lehnhardt, and of course also all the colleagues that were somehow involved in the work from Miltenyi, and I want to single out especially Olaf Hart, Stefan Tomiuk and Laura Nadine Küster, without whom this work would not have been possible. With this, I'm coming to an end. I'm also say thank you to the audience for your attention and interest into the topic. With this, coming to an end, I hope you liked the presentation, and maybe later in the Q&A session we can get in touch with each other. Thank you very much.